Thank you very much for this nice introduction, and thank you for having me here in this beautiful city and this fantastic campus. The title of my talk is The Complement System, a Novel Sculpture of the Developing and Injured Brain. And as was mentioned in the introduction, I started as immunologist studying the complement system. In the process, I made a couple of knockout mice, which I thought would be useful to study the role of complement in different diseases. And I found that the brain is actually the most interesting organ, even for immunologists. So that's what I'm doing now. And to give you just a brief repetition of what the complement system is, in case you missed that class in microbiology or immunology when you went to college or university. So the complement system is part of the innate immune system. That means it helps us to eliminate pathogenic bacteria. But it has a lot of other functions, not least in the brain, and that is what I would like to convince you about in this talk. In some way, the complement system resembles the coagulation cascade. That means that there is a whole bunch of proteins. Many of them are precursors of proteolytic enzymes, then activate the next protein in the cascade. So it's a cascade of interactions where the central molecule is the one indicated here, abbreviated as C3, which stands for the third complement component. It can be activated in a number of different ways, depending on the triggering event of the cascade. And the proteolytic cleavage of C3 then generates two fragments, a larger fragment which is called C3b, which functions through receptors called complement receptor 1, 2, 4 that are mainly involved in some aspects of phagocytosis, or C3b is involved in the amplification of the cascade and the activation of C3. C3a is a smaller fragment which acts through its own receptor, which I abbreviate here as C3ar. So just briefly about what the larger fragment C3b does in the brain. We have contributed to this work in my lab partially, but the main bulk of research on C3b in the brain comes from the lab of Ben Barres and then Beth Stevens, who showed that C3b functions as an opsonin of weak synapses in the developing brain. So it tags the weak synapse, and in this way, the synapses can be then phagocytosed by microglia, who are the only cell in the brain that express the right receptor for this. And this elimination of weak synapses or synaptic pruning is important, for example, for the segregation of the signals or inputs from the two eyes in the lateral geniculate nucleus in the thalamus, where the cells in the thalamus initially receive input from both eyes, but through the process of synaptic pruning, then maintain only input from one eye. And this process of synaptic elimination then can be activated later on, and it has been associated with neurodegeneration, such as Alzheimer's disease. But I would like to focus my talk on the smaller fragment, C3a, which as I mentioned, functions through its own receptor called C3A receptor, which is G-protein coupled receptor that in the rest of the body is primarily expressed by white blood cells, by leukocytes and mast cells, and functions as an important mediator of inflammation, leading to recruitment of white blood cells to the site of injury or infection, and activation of these cells. And this happens also in the brain in the context of injury in the acute phase. This complement or the C3A receptor is also expressed on more or less all the cells in the brain except for oligodendrocytes, all the cells including neuroprogenitor cells, neurons, astrocytes, microglia, endothelial cells, as well as the epithelial cells in the choroid plexus express this receptor. And there has been intense research in the past, say, 10, 12 years, showing that there is a range of functions that the activation of this receptor leads to. And one such important function is the regulation of migration of neurons in the developing brain. As you probably know, in the developing brain, neuron progenitor cells are located around the ventricle, in the ventricular zone, where they proliferate. And then the progenitor cells or the developing neurons migrate along radial glia towards the cortex, towards the cortical plate. And any impairment in this migration has been shown to possibly lead to intellectual disability or psychiatric conditions such as schizophrenia or autism. So it's an important process. And interestingly enough, your colleagues, not from Jerusalem, but from Rehovot, group led by Orly Reiner, recently showed that mice that do not have C3, that is the precursor protein for C3A, or mice that have knockdown of C3 in a particular time at the development between the embryonic day 14 and 18, they have impaired migration of neurons towards the cortical plate. So this is, here is the ventricular zone, this is the cortical plate, and this is how the labeled neurons look like in a wild-type mouse. But in a mouse that doesn't have C3, these neurons are all over, then don't make it properly to the cortical plate. But if they treat it, or if they inject it, the embryos with agonist of the C3A receptor, or a combined agonist for C3A and C5A receptor, they could actually rescue this phenotype. So this is a very nice study that shows that complement, and in particular C3A and C5A receptors, are important for normal neuronal migration in the developing brain. And then they went on to show that in autism spectrum disorders, patients with spontaneous mutations very often exhibit mutations in complement genes, not only the gene coding for C3, but also in, for example, C complement receptor 1, in this gene ITCAM, which codes for part of the phagocytic receptor for C3b, and a number of other genes. So these findings from Orly's lab may have very important clinical implications. Now, I would like to talk about research from my own lab, where, as I mentioned, we were interested in what complement does in the brain. And we started years back by exploring the possibility that complement can have something to do with regulation of neurogenesis. So we used mice that do not have C3, and we looked at the generation of new neurons. Here you see the labeling of cells with a double coating of the newly formed cells with the double coating in the hippocampus, the subgranule zone, and the subventricular zone, and then the terminal destination for these neurons, the olfactory bulb. And you see that the mice that do not have C3 have reduced numbers of these newly formed neurons in both the hippocampus and the olfactory bulb. And the same is true for mice that were treated with an antagonist of the C3A receptor. We saw the same when we look at more mature neurons using antibodies against nu-N. Again, there was reduced generation of neurons in the hippocampus as well as in the olfactory bulb. And then we went on to confirm the role of C3A receptor signaling in this process by using mice that do not have the receptor, and we saw the same. So these told us that signaling through the C3A receptor is important for normal generation of new neurons in the two neurogenic niches under basal conditions. And we wanted to confirm these data by in vitro studies, looking more into the mechanism. So we then used neural stem cells or neuroprogenitor cells that were derived from adult mouse brain, and look at the effect of C3A and C5A on the generation of new neurons, and so that the C3A increases the production of new neurons in these cultures. It also stimulated the maturation of these cells, here assessed as neurite outgrowth. But we didn't see any effect on the proliferation. And interestingly enough, in the same study, we found that C3A indeed affects migration of neuroprogenitor cells. It did not have that effect on its own, but it modulated migration of these cells when they migrated towards the gradient of stromal-derived factor 1 alpha. So if the concentration of SDF1 alpha was too low, then C3A increased migration of neuroprogenitor cells, but at rather high concentrations, it reduced it. So it seemed to us that it works like a fine tuner of migration of these cells, which in these experiments more look at the role of C3A under inflammatory conditions or after injuries. I'm not saying that this is exactly the same that what's happening in the developing brain, but it points in the same direction. We were also interested in the possible effects of C3A on astrocytes, and since the interest of the lab is ischemic brain injury, we looked at the effects of C3A on astrocytes that are exposed to ischemic stress. And as you see here, the C here means control conditions, and I is ischemia. When we looked at cell death, we saw that C3A had a protective effect on astrocytes in wild-type mice, but this effect was lost in mice that did not have, or not mice, but cells that were derived from mice that do not have the receptor. And this is the same experiment with another model of ischemia that we used. And this is just a confirmation that the effects of C3A on astrocytes are mediated by controlling the phosphorylation or activation of caspase 3. Now, I would like to have or to turn a bit more clinical in the rest of the talk and tell you a bit about our studies on what complement does in the ischemic brain. And the main condition that we have been focusing on is stroke, which, as you might know, is a devastating condition that annually affects about 15 million people worldwide. About 1/3 of stroke patients actually die within the first months, and 50% of the survivors are left with a permanent disability, which, depending on the location of the infarct, may mean affected or reduced ability to control the movements of arm or leg, or problems speaking, swallowing, disturbances of vision, problems with balance, and so on. But importantly, stroke not only leads to the loss of brain cells in the ischemic area, but it also is a very powerful activation of neuroplasticity, which in many ways are the mechanisms that we use every day when we adapt to the changing environment and when we learn new things. So we now know that after stroke, the surrounding regions of the brain and in some cases also quite remote areas of the brain respond by increased generation of new synapses, increased activity of synapses, rearrangement of the connections between neurons. In the peri-infarct area, also generation of new blood vessels, generation of new cells, including neurons. So all these mechanisms of neuroplasticity are extremely important for, or the basis for the functional recovery, at least partial functional recovery, that we see in many stroke survivors. But interestingly enough, there are huge differences in the ability of stroke survivors to regain the functions, to stimulate this plasticity of the brain on their own. And therefore, one needs to find ways to stimulate these processes in those who do not have this ability so much on their own. And in line of this research, we thought that the complement system could be important. We started years back again by looking at or by using the mice that do not have C3, and we saw that these mice in two different models of ischemic stroke had somewhat larger infarcts, which would show that complement may be neuroprotective. We found that these mice in the subventricular zone, which normally responds to stroke by increased neurogenesis, increased generation of migrating neuroblasts, that these mice had impairment in this response. And they had also reduced generation of new neurons in the penumbra, the region around stroke. So that was very intriguing, and but we thought that based on the findings from the neurogenesis studies, complement might, or C3A receptor signaling might have a role in neuroplasticity after stroke. And to investigate that, we used, again, mice that do not have the C3A receptor, and we used also mice that were originally generated by the lab of Scott Barnum in the States, which express a large amount of C3A in reactive astrocytes. So these mice we called GFAP-C3A because the expression of C3A is driven by the GFAP receptor. And then, of course, we used the respective wild-type controls for these mice. And when we subjected these mice to a model of stroke, which does not really generate any markable penumbra, we saw that there were no differences in the infarct volume. And that was good, that was actually important for us, because if we wanted to study the neuroplasticity responses and recovery afterwards, it was good that we had the same starting point for all our groups. And we saw that in the mice that do not have the C3A receptor, there was reduced generation of new neurons in the peri-infarct region, and the opposite was true for mice that overexpress C3A, pointing to positive role, again, of C3A receptor signaling in this respect. But even more importantly, I mean, I don't want to say here that the generation of new neurons is the main contributor to plasticity and to functional recovery after stroke. I'm convinced that there is much more to it, as I mentioned before. Therefore, we looked at other markers or other parameters of plasticity in these mice. And one of them is growth-associated protein 43. So we used antibodies against GAP43, and then they did high-content image analysis of the images in the peri-infarct region, in different parts of the peri-infarct region. And so something similar that we saw when we look at neurogenesis, that again, the mice that do not have the receptor had reduced expression of GAP43, and the overexpressing mice had more. And the same was true when we looked at markers of synapses, and in particular markers of excitatory synapses. Again, signaling through C3A receptor seemed to stimulate synaptogenesis in the peri-infarct region. So that was very positive, but it's not really clinically relevant. So we thought of a way how we can make this more clinically relevant. And an interesting approach to us appeared to just see if we can take wild-type mice and give them nasal drops with C3A, if that would have a positive effect on neuroplasticity and recovery after stroke. So we did that. We also first confirmed that if we give C3A to mice in this way, that we can actually get it to the brain and we can detect it there. So these images, they just show how C3A that is tagged with a fluorescent label can be detected in the brain of naive, non-injured mice 1 hour or 3 hours after, and these are the brains that we then took out, where you see that it's much clearer. So we can get C3A into the brain, and we can detect it there for at least 3 hours after the treatment. So then in the stroked mice, we thought that for this treatment to be really relevant for the majority, or if not all stroke patients, one should wait until the post-acute or chronic phase after stroke. So that's why we waited for 1 week before we started with this treatment with nasal drops containing C3A. And so you see here the treatment did not have any effect on infarct volume, and that was what we expected. But it increased the expression of GAP43 in the peri-infarct region of the treated mice compared to mice that were treated with just the vehicle. And it also increased the density of excitatory synapses in the peri-infarct region. But did it have any effect on recovery? So to do that, we treated the mice for 3 weeks after stroke, again starting 1 week after. And we used 2 tests that assess the motor function. And as you see, so there was a grid walk test where we see how many fold steps the mouse makes with the affected paw. And rearing in a cylinder where we look how much the mouse is able to use the affected paw to support itself when rearing in a cylinder. And in both tests, the mice that were treated with C3A showed faster recovery that was maintained even after we terminated the treatment. As you see, we treated only for 3 weeks, but then we followed the mice for several weeks thereafter. So now in the last part of my talk, I would like to talk about another model of ischemic injury to the brain, which is then a condition that is called hypoxic ischemic encephalopathy, which is a major contributor to perinatal mortality and long-term neurological impairments, such as cerebral palsy, epilepsy, or cognitive impairments, or hearing disabilities. This hypoxic ischemic encephalopathy is a consequence of birth asphyxia, but also, for example, impaired respiratory function in the newborn. It can be caused by cardiac disease or head trauma, and affects about 1 to 2 term babies out of 1,000. So we used a model of this hypoxic ischemic encephalopathy, where we occlude the left common carotid artery, and then we put the mice into a chamber with reduced concentration of oxygen for 30 minutes. And then we follow the mice at different time points after for different projects. So in this particular one where we started, we used wild-type mice and mice that express C3A in reactive astrocytes. And when we looked, I should also say that this injury in mice is done on postnatal day 9, which in terms of brain development is comparable to the newborn in humans. And then we looked at these mice when they were 1 month old, and so that the mice that overexpress C3A, that they had larger hippocampus or reduced loss of hippocampal volume due to this hypoxic ischemic brain injury. So that was positive, but again, not clinically relevant. So in the next experiment, we used wild-type mice that we again subjected to this hypoxic ischemic injury. And some of them we treated by a single injection into, with C3A into the brain ventricle 1 hour after the insult. And when we looked at the mice when they were 2 months old, we saw and assessed their cognitive performance. We saw that the mice that were injured but treated with PBS had reduced learning and memory. These are the controlled mice, and these are the mice that were treated with C3A. So this showed that C3A in wild-type mice can be used as a treatment to ameliorate this cognitive impairment induced by hypoxia ischemia. And when we did that in mice that do not have the C3A receptor, then the treatment had no effect. So this really showed us that it's the C3A signaling through the C3A receptor that is responsible for this positive effect of the treatment. Now, again, to be more clinically relevant, we then went on and tried the nasal drops containing C3A in wild-type mice. Here we used a different test, but the conclusion from these experiments was the same, that we don't have to inject C3A into the brain, we can give it in nasal drops, only 3 times, starting again 1 hour after injury, but then continuing for only 3 days. And when we test the mice as adults, they have better cognitive performance than the mice that were treated with the vehicle. So in conclusion, I believe that the complement system can be one of the potential approaches to improve recovery after ischemic injury. I mean, it's not the only one. We know that we can stimulate neuroplasticity, for example, by interventions such as horseback riding or rhythm and music therapy, by dancing or imaging dancing, but a combination of pharmacological approach with this multimodal stimulation or rehabilitation may be the key to the optimal recovery after ischemic brain injury. And perhaps agonists of the C3A receptor can help us in that direction. And on the last slide, I would like to acknowledge all the people who contributed to the work, in particular Anna, who was the driver in the stroke experiments, Javier, who did the hypoxic ischemic injury studies, Henrik and Corina, who helped us to set up the hypoxia ischemia model, Milos and Michael. Milos, who is here in the audience, who is a great collaborator in many ways, and also my husband, so I have to acknowledge him, and our clinical collaborators. Thank you very much. I'm happy to take questions.